What we'll do now is give you a bit of an overview of the national drug discovery, on quite a number of aspects, information on how, what it is, what's its aim, how you will be able to access it, the capabilities, and Helene will talk to you about that aspect, and how to apply and how to get in touch. So I think the key message here today is really get in touch with us, really communicate with us and see how you can start your journey into the drug discovery path. So one of the key message really here, and as many of you know, is really to try to fill a gap in the drug discovery path, and really this link, as was mentioned several times this morning already, this link between the academic funding, the basic research, which really is where discoveries of new targets happens, these weak links for diseases is happening, and how we can bridge that to a translational path to link to pharma, biotech, and really enable this drug discovery pipeline. So this is what we are trying to address with the National Drug Discovery Center, and further to link into other opportunities to do medicinal chemistry and develop the molecules further down the line. So that's where we're located. So many of you would know this drug development pipeline, but it's just to remind you that we're going from target discovery, which basically is really the fundamental research that happens in the precinct in the nation, where these new targets for new disease are discovered. And then there is a process that goes along these different steps between hit generation, hit confirmation, which is really what the National Drug Discovery Center will be about, but then goes on into hit optimization, etc., where there's a lot more medicinal chemistry that happens, engaging a number of organizations already in place. And then all leading towards the clinic, with preclinical and clinical studies. So these are really, broadly speaking, major steps in the drug discovery pipeline. As was mentioned a number of times this morning already, we obviously have a lot of experience at WEHI in drug discovery. The center, or the drug discovery infrastructure, was established under Suzanne Corey's leadership at the Institute in 2000-2001, with the first high-throughput screening lab, medicinal chemistry and structural biology, that really supported all the activities that we had. We've had since more than 100 collaborations based on this over the last 10 years, academic, pharma and biotech, and multiple high-throughput screening, whether they were originating from projects at the Institute, but also around the precinct and even with pharma. So the message really is that we're not building this National Drug Discovery Center out of nothing. We really have a lot of expertise, and the idea was really to provide that to the community in an expanded facility. Again, the story of venetoclax is here. You all know about it, but really this provides a blueprint about what we want to do is really taking these really key groundbreaking discoveries and to enable their translation into small molecules. It took about 30 years from start to finish with venetoclax, and part of that is also in the basic research to a better understanding of where we need to target and do the drug discovery work, and that takes time in itself. But with the National Drug Discovery Center, it's really to accelerate the first step of translation, to make sure there is less gap between the critical discoveries and the translation, and to make sure that a lot of the value is actually developed here in Australia rather than overseas. So as we were also contemplating internally to expand on our activities in drug discovery, we also built a business case to actually offer this facility and expertise and all the people around the screening facility to Victoria and to the nation. So that really, with the aim of accelerating this translation and creating a hub for drug discovery, for screening and drug discovery here at the Institute. Obviously, that's really with the aim to also strengthen the network of facilities that are along this pipeline that I showed you, and really enable that translation. So that really led to the development of business cases to federal and state government to really expand this facility further. The results was major fundings, obviously, as we've mentioned already, WEHI plus philanthropy really contributed in a major way in establishing the facility, in its infrastructure and instrumentation. The federal government provided essential funding to actually run the facility, which will support people around the facility to really run the screens and help people from outside the institute to have their science translated. And more recently, the Victorian government provided further funding into additional instrumentations, which will really help extend the type of assays that we can run on the platform, in particular in relation to biology that's developed here on the precinct, but also established a better compound management facility here, which will further accelerate the process of screening. And we also have protein production, which is a really essential part of the screening part, where we need to have enough proteins to run these screens. So where does the National Drug Discovery fits in that pipeline that I showed? So it's really here to offer the hit generation and hit confirmation steps in the process. And just to go a little bit more details, to do this translation, this very early step between the basic discovery and the identification of what can be the start of a drug discovery project, there's a number of steps that are shown here on the bottom of the slides. The assay development is the transition between the research lab to the screening lab. And then assay transfer is when you actually, once you have developed an assay that works for a robotic platform, so you've chosen a readout, you know what type of experiment you're going to use on the platform, you transfer that onto the robotic platform. And that will require miniaturization, optimization of reagents, etc. And so that's a very key part of the process. Then we will run a pilot screen, which is really here to test how this assay is performing on the robotic platform and looking at statistics, etc. And then we will run the full deck screen with a number of assays decided earlier on to decide how we're going to remove any compounds that could interfere with the technology and etc. That would then lead into medicinal chemistry. So what the National Drug Discovery Center will offer is really these three key steps in the process, here shown on this slide. And here to just give you some additional information. So what we will require from you and the community, the scientific community, is an assay that has already been developed and is ready to be transferred on an automated platform. And we will, part of the application process for this, for the National Drug Discovery Center, is to provide you with the information on what this assay can look like. And obviously, as I said right at the start, is to help you reach that stage with communicating with the screening lab. At the end, you will be provided with a list of hits, which are these small molecules that are showing activity in the assay that was developed and run. And then you can decide where you want to take your project from there, especially in terms of the medicinal chemistry, which will really start creating or going forward the drug discovery path. So this is the part that we've decided to also provide you with this and stop at that stage where you can decide where you want to do the medicinal chemistry part. So I will hand to Helene, which will describe in a little bit more detail the technique, the technology part, and then I'll come back to address a few other points around applications.
Thank you, Guillaume. Hi, everybody. So I will go into more technical detail here, just to tell you how we are going to run the screen and what we are going to do. So the first step would be, as Guillaume said, the pilot screen. So we test a subset of the library to make sure that the assay is robust enough and can be then further screened. Then the next step is to run the primary screen, where we run the full deck screen for 300 maximum or 300,000 compounds. So it's the compounds are tested in one concentration, one single point. So that's why it's extremely important that your assay is robust, because it's a one-shot test. Then we select the hits and further confirm them, and also remove the false positive by running a counter screen assay. And then finally, we titrate the compound in a dose response assay to identify the potency in both the primary assay, but also the counter screen assay. So the center relies on 4 different pillars. First, the team, the library, the small molecule library, the software and database, and then the robotics and instruments. So first, the team. I think this is the biggest asset of the center. The team it has more than 100 years of combined experience. To be successful in screening, it requires a variety of different skills. So we have different experts in automation, engineering, data analysis, assay development, and screening that are coming from both academia and industry. So the center and the lab has extremely expanded since 2014. So we were 4 in 2014, and now we are 14, and we will further grow in the next few years. In terms of technology, what can we screen? So this is a table showing you the different types of assay and classified by a target class. And with, at the bottom, the different assays and type of assay that we can run. So the take-home message here is that we will be able to screen all type of target class. In this table, you can see in orange the recently implemented technology. In yellow, the long-standing technologies that we've used many times. And in red are the new technology that we are going to integrate in the coming years. So how do we read out? So how do we read those technologies? So we have 3 different types of readers. We've got a high content confocal microscope and automated flow cytometry for phenotypic screens, so for cellular screens. But we are also running biochemical screens with a multi-mode reader able to read fluorescence, luminescence, FRET, fluorescence polarization, and so a lot of technology. So if you've got any question on the technology that we have and what type of tests that we can run, please do not hesitate to contact us. So those readers have been integrated on 3 different type of platforms. So we've got a biochemical platform. So it's integrated all the different instruments all together with a robotic arm in the middle. And what you can see here is that the readers are on carts, can be undocked to the station and docked to other stations. So it's a highly modular system because we can then dock those readers to our other platform, which is the biological platform dedicated to cell assay. So the same kind of platform, but with an automated cell culture liquid handling system here. So again, it's a really a modular system. We are optimizing the equipment usage here. We can customize the platforms, and we also can add more technology if needs be, because it's a scalable integration. This third platform is the satellite platform, and this one is used for the assay transfer. So when you come to us, we will miniaturize your assay on this small little platform here. So your assay, so you will come with us with a developed assay, and the collaborator will provide specific materials to run the assay, such as cell lines or protein, but it will also have to meet a specific requirement to be eligible and to be fit for essay transfer. So you will have to demonstrate that the essay is working in a 96 well-played format with a minimum robustness and signal to background, and that the reagents are stable enough to be run on the platform. So if you need more information and if you want to have more information on the requirement, eligibility requirement for your essay, please go to the National Drug Discovery Center website. So what we offer our expertise, so a fully staffed facility. We also offer, as I've just presented you, the latest cutting-edge robotic instruments, and the researcher from eligible Australian institution can apply for a subsidized screen at 90% discount. So what will you get? You will receive a detailed report with the standard operating procedure of how we've run the screen, exactly what we've done, but more importantly, the screening result, which would be a hit list with obviously the compound hits with its structure, the potency with the IC50 data, and some specific data like the images. You will also have the supplier information for you to purchase the compound. So with vendor catalog and catalog ID. And what I want to highlight here is that the intellectual property, if any, is fully owned by the collaborator. So now I will hand over to Guillaume to discuss about the costing model and the application process.
Thank you, Helene. So I'll just give you a bit more information to conclude the presentation. So in terms of the cost, we've given you here an idea of the cost of a full campaign, which is quite significant, and that's where the help, the support from governments here is absolutely essential, and therefore it's heavily subsidized price that is offered to the community here, which will be about 10% of that overall cost here, so between $30,000 and $45,000. Half of the fee will be paid upfront, payable upfront, and then depending on the cost of the actual screen, then the rest will be then paid at the end of the, upon receipt of the final report. So how to apply? So we've set up a website, and with all the information, the criteria that Helene was mentioning just before in terms of how the assay should look like and the parameters of this assay. You will see also the application, and I'll talk a little bit more about the application itself and what's important to apply about the type of project that would be eligible. You will be able to request a login and you will have access, and there is a specific portal through which you will be able to apply that's been developed and is now accessible. So how are projects selected? So that's a very key question. We've set up an expert committee panel with really expertise from fundamental biology, translational biology, all the way to drug discovery, to really be having a great body of expertise to decide on which project should go into the National Drug Discovery Center. And you will see, I will show you the list. It comes from experts throughout Australia, from an international research organization. So one of the points that I'll re-emphasize later is that we obviously want to enable translation. It will be also to develop tool compounds that can help you with your biology. So really what will be very important is the science itself and the potential for developing a tool compound, but also a drug discovery project. The National Drug Steering Committees here, you will see that it's mainly, the majority are from outside WEHI, and they are representing a number of states, and as I said, a large range of expertise, really to select the best project coming into the National Drug Discovery Center. So the project selection, the key selection criteria will be really about the science itself, scientific quality and rationale, the feasibility of the screen and the innovation of the approach and of the concept around the target that you are working on. We are ramping up the activities in the center, so we will select by the end of this year about 2 projects. It will ramp up to 4 in the second round, and then capacity to grow to up to 16 screens per year. In terms of timeline, so really, we are really opening the center today, and you will be able to apply directly and start communicating with us about how to apply and what in your project and the essay that you want to develop for this. You have the key dates here. The cutoff date for consideration for round 1 will be the 7th of November of this year. The expert panel will meet early December to select project, and they will be announced early next year. And then further along, expected cutoff date for the round 2 will be around March 2020. And we're expecting to have probably two rounds per year for accessing the platform. In terms of confidentiality, all the information that you're providing to the National Steering Committee for the selection will be obviously treated confidentially. We have put in place a whole process and a documentation and legal background for this to be confidential. As we've said several times, please do visit the website to get all the information and to also download the mutual confidentiality agreement template. And then before you submit any information that you think is confidential, please sign and submit this before the application. There are 2 sections in the application that are non-confidential that we will use for presenting the type of projects that have gone into the platform. It's the title and the non-confidential life summary. So there's a number of contacts here. You will find some documents and information outside the room, but please do not hesitate to contact us at any point and as of today to talk about the translation, the project you have, and how we can help you in that journey. So really, the key message is if you've been working on a target and this target is going to be important, or you want to have a small molecule that targets this particular protein, or if you have a cell line that really informs on a very important biology, novel biology, and you want to also develop compounds, or as you will see here, we're interested in both targeted protein-based assay, but also in cellular assays. Please contact us, and we'll help you start on this journey. To conclude, and that will open questions, and later on also with the morning tea, you don't hesitate to ask questions to Helene, other members of the National Drug Discovery Center, or myself. But I just would like to thank the funders and donors that have really contributed to where we are at today with this really expanded facility. And I thank you for coming, and hope that and welcome any question. Thank you very much.
Very exciting development. 2 questions. The compounds in the library, when they come out as hits, do they come out annotated with past campaigns for specificity against other targets, or is it just a very simple result? It's quite narrow.
So I'll first answer, and if Helene wants to. So some of the targets we've been working on obviously are confidential, so we probably won't be able to disclose a lot of of that information in particular and again, the targets that you will provide will also be treated confidentially, so we're probably not going to be giving that information. Just to emphasize, we're not just going to give you a straight one data point out of the primary screen. There will be titration curves There will be some counter screening as well, at least on the technology side. And also, if decided early on, any other targets you want to test the compounds against to look for specificity, et cetera, et cetera. So.
Yeah, if I can just comment, so we won't give any information of other targets that have been screened, but we can give you information whether the compound is a frequent hitter or not. So just the percentage of time this compound has been hit.
Yes, and 300,000 compounds is a lot, but is there a provision to bring third-party libraries into the platform for additional breadth?
Yeah, that's possible, absolutely. That will change, obviously, if, depending on the size of these additional libraries, that could change the overall cost of the screen, but absolutely possible, yes.
It's a great initiative, Guillaume, and congratulations to you and WEHI for really driving it. I just had one question in regards to the subsidy. So it's been mentioned a couple of times, there's a 90% subsidy for accessing the screening capability. Is that available just to academic groups, or is that available to SMEs and for-profits as well?
So we're still working on the model here. What we can say is definitely all academic institutions or laboratory from academic institutions will be eligible, and technology transfer out of these universities. And on the other end, who will not be eligible is big pharma. However, we can run the screen, but that will not be eligible to the actual subsidy. So we're still working the various eligibility in the middle, but we want to obviously support as many projects as possible in that space.
Thanks. So you alluded a bit through your presentation, what might come next. So you have a hit, you're a lab maybe that has no experience or abilities. How can you turn a hit that is potentially still low affinity, low potency into something that might actually become a useful lead?
Yes, that's a very important question. So the decision really, we decided that WEHI wouldn't become an expanded center for offering the medicinal chemistry part. However, we really want the center to be integrated in a network of capabilities around the nation or locally that can help you progress this. So if you don't have the capability in your organization, we will be here to point to you to organizations that can help you, whether they are here on the precinct or nationally. So that will really, that's a key point, that we want to have this facility embedded into a network of drug discovery facility on the path towards yes.
Just to follow up that previous question, you might like to indicate the cost involved in going forward with the hit. I know that's a difficult question with no single answer, but it's part of the journey that people will be interested in figuring out how they can manage.
Yes, so that's a very important point. So it will depend largely on how that next step happens and who is partnering with you with your project to help the next step happen. So hopefully if you find the right partner and the right organization and the right model, then that will also help you really do that, because I think what Peter Coleman was trying to say is that it can cost a lot of money to do the next step, but there are a lot of organizations that are here to take on your project based on your science and the hits coming from a facility like this one to really support the next step of the drug discovery. Okay. No, Nick?
So there are many. So I guess there are many hits which a medicinal chemist would look at and say, nah. Is there any information that comes out of the hit that talks about how good these might be as future.
So we've discussed that internally a lot to actually discuss whether we should provide advice on which one we think are better than others. But as you may know, medicinal chemists have different views on the hit list, and we've preferred, we can help and guide that way, but we prefer to actually officially let the person that brings the project decide what they want to do, so we don't bias their view on the hit list. If there are really red flags that we absolutely know for 100% sure they shouldn't, they've come through all the confirmation and counter screening. If ever that happens, we will be here to say this came through, but we really think that's not going to be productive. But otherwise, we'll really we'll leave the lead scientists decide how they want to have our input or not, or how they want to proceed with this. Okay, thank you very much. So please, thank you again for coming. We hope we'll be working with all of you in the next future, and then please join us for a morning tea just after this. Thank you.